Hi, and very good morning to all of you. In the session 9 and probably the last session of this entire series, we'll be going to discuss about the systems biology. That is, we have been learning so far that the complete biological all the analysis is very complicated. We have seen so far from genomics, proteomics, and currently the metabolic networks. We have seen numerous molecules are constantly interacting within a cell, among which, if I am able to analyze that complete molecule as a system, just like the flux balance analysis we did, okay, we are considering all the metabolites together to predict or analyze the flux balance per unit time within a cell. Okay, so that if I am calling it as a system, so the entire biology or the biological complete analysis is termed as systems biology. Okay, for which a language like HTML has been developed. That is termed as Systems Biology Markup Language. Okay, the SBML. And moreover, a tool or different algorithms have also been developed for analyzing the data. The one of them is mentioned is Systems Biology Workbench, SBW. See, these are very complicated parts, but people have been working out extensively for numerous things. Okay, all right. So, what are biological systems? We have been seeing that the complex systems emerge from the properties of the molecular networks of simpler elements. We have always been seeing that. Okay, in the metabolic networks. All the complex metabolic networks have been there, and we are trying to truncate the required segment of the metabolic network to analyze it further. All right. So the biological systems that functions that rely on the combination of the network and specific elements involved. Okay, as obvious. So all these parts, whenever we are doing all this analysis, what we need to do first, we try to like data handling. We extract all the data required part. We do everything. Okay. Once we get the required data, as we have been discussing in the last session, we have been discussing about network pruning and others to extract the required data. But all that comes under data handling. Once we need to build up the required data. Once that is built, we try to model the required part, required metabolic network. Okay. What is meaningful for us? All the outlier solutions we discard, just like we have seen in the case of network part. All the outlier like networks coming up or the solutions we are discarding. Once that is done, then you try to build up an experiment. Okay, you try to generate the experiment. This is the complete vector experiment based on all the predictions, all the data coming out here. Okay, you try to get the data, build up the network. Once the network is built, okay, you try to analyze it with experiments. And once the experiment finalizes or proofread that this metabolic network is correct, only then you affirm that this results or these predictions are right. And if something is wrong there, what it means? This means that either you have missed some data, some important crucial element is missed out here, some crucial metabolite which was required. Okay, you missed out completely. Or you have considered some additional useless data there, which was trivial, which was not at all making sense, but you just considered it. Okay, so you need to recheck the initial data set. So the data handling approach comes up again here. Okay, and it goes on as a complete cycle. Okay, it goes on as a complete cycle. Okay, so that you can improve the accuracy. All right. So this complete part involves several academic fields: biotechnology, biology, statistics, physics, chemistry, information science, and computer biology, computational biology. Okay. And what is all it giving us? Improved biological knowledge, new experimental techniques in genomics and proteomics to validate the required data, improved mathematical modeling of biological processes so that we are able to improve the required predictions. Whatever we are generating is exactly true. Computational resources for the simulation of complex systems. People have been writing the very complex codes, and they are able to build the tools which biologists could not even dream of. And all these tools are helping the biologists to work out the things better way. So the computational biology and the biological experiments go hand in hand to get the required data sets. Storage and retrieval capability in large data sets and data mining techniques. People are building better data mining techniques. So from the billions or TBs of data, they are able to get the required data. And that too in fraction of seconds. So building up some better arrays, better it's all possible because of high bandwidth of internet. So people are deploying each and every aspect. But what is the ultimate goal here? Goal is to build a reliable model of the biological systems. Okay, and attempt to reliably describe the biological mechanism. From whatever the data set you consider, whatever the things are there, you try to build up a mathematical model, probably the best model ever built from that data set. That is the ultimate goal. Okay, and if some assumptions you are taking, you need to validate that are they credible. What is the reliability of all these assumptions when I'm initially building the data set? Because I've been discussing that if the data set is incorrect, either some element is extra or some element you have like should not have considered, but still it is there. So you need to discard it. Okay. Build prediction tools to design cells and desired properties cheaply and reliably. Like many tools like virtual cell, e-cell have been developed, wherein you just give glucose to the cell in the computer and the cell will behave the way it should do in the lab. So you can exactly quantify all the log phase, lag phase, and various other biochemical and physical properties in the computer itself, rather than doing the things. Correct? Because we cannot do a WhatsApp message to the cell. What is the fate of some molecule the moment I'm giving the input of some other molecule? Okay? So these kind of tools are very important for us, especially when you are considering large number of cells, large number of metabolites involved, and the system is very complex. Okay? For novel drugs and therapies, especially when you are analyzing some drugs or viruses. Okay? Study the mechanisms responsible for the altered phenotypes. Okay, so these are the key goals. So now let us understand the systems biology in more detail. It is a study of mechanisms underlying complex biological processes is integrated system of many interacting components. The many interacting components are there. It involves a collection of large set of experimental data. Okay, was all of them got different results have been published in various literature resources. So the moment you are considering something, you need to consider all the published data set as well. Okay, the expression values, all the mutation diagrams, mutation analysis, and every single thing. Proposal of mathematical model that might account for at least some significant aspects of this data set. Okay, you need to build it. So once you correct the data set, then you need to build the required mathematical model. Building the accurate optimal computational solution, okay, of the mathematical equations to obtain the numerical predictions. We need to get it so that we get the best model, we get the best computational solution. Okay, and then we lastly check assessment of the quality of the model by comparing the simulated data with the experimental data. Okay, which you may need to perform in the lab. Okay, and all this go again and again iteratively until we reach the required score, required accuracy. Okay, so now when you are building the mathematical model here, what does the mathematical model actually mean? See, as we have been discussing, this systems biology is a very complex part. It involves numerous scientists. Okay, it involves numerous scientists, both from mathematics, computational biology, chemistry, thermodynamics, and all of them. So for each of it, the model means something different. but all of them are interrelated. For a computational biologist, model just means how well a data is simulated by the given set of parameters. That's it. So if my computational algorithm is able to give me the required scores, the model is best. Or if something abrupt is coming up, means some bugs are there, or the computational model is not yielding me the required results which I should expect from the biological system. Geneticist, it means the finding the correct genotype and phenotype for a given condition. That's what the model means. Okay. Now for chemist, it just means the what is the like reaction network. That's it. What is the metabolic network altogether? Define molecules defined by different reactions. So it just means the complete metabolic network or the reaction network. It has nothing to do with any other things. For mathematician or engineer, this means finding the reaction network by a system of linear or nonlinear equations, which may be deterministic, which may be stochastic equations, but anything. But mathematical engineer means try to build the equation based physical part. But now you understand, like, for actually building the required program, I actually need all of this. Okay, we need all of this. Then only the things can be rightly worked out. Okay, now see the part. How to integrate the data. Observation of biological phenomenon is described by the accuracy of the available experimental techniques. Okay, a strong impulse to the development of systemic approaches in last year has been given by the high throughput technologies for genomics, transcriptomics, proteomics, like the things have been working out different part and they have resulted into large amount of data. So all this data is linked together. As I've shown this diagram earlier as well. All this data is linked together to get us the required output. So data integration is the key step here. Then we build up the model. Okay, we try to build up the required model. Okay, interaction map and kind of interactions, experimental data, checking up each and every part. We try to build the model. Then, after building the model, we like a reliable mathematical model. We try to put the mathematical equations as well. Okay, the choice of mathematical model. But see this line importantly. The choice of mathematical model or an algorithm depends on the problem, the purpose, and the intention of the researcher. Okay, if he is interested to get the best solution, he'll try to do iteratively and not just once. Okay, as it should be there. Okay, so different models can provide same information in different aspects, but we need to consider each and every single aspect. So putting up all the data available and putting up all throughout from different perspective, we need to get the best possible model. Something which is able to put the required data rightly. It's the best computational model. Like whatever the input I'm changing, whatever the thing I'm changing, my computational model should be able to give me the required result. Something which should actually happen in the cell. Okay, and what is the impact of mathematical model here? It should be able to predict the things the way it should be. Okay, so the flux balance analysis and all those things, like how much time the system, the Excel should take to give the required result. 5 mg of a reactant becomes 1.4 mg of product. In how much time? Exactly the same time it should my cell should take in the computer to give me the required result. Okay, that's the impact of mathematical model here. So see, the model development is very crucial here. Okay, so see, iterative refinement of hypothesis is required to improve accuracy. Okay, selection of model structure, level of description, stochastic, deterministic or stochastic model, discrete or continuous variables. I'm sorry. All this just means how many variables are considered in the model, like are the constellation constantly changing throughout, or like that is continuous, or like if their constellation is only changing when needed. They are discrete. They are not dependent on each other. Okay, are the complete model structure, is it static? All the molecules are itself alone separately. Okay, or dynamic, see, or spatio-temporal dynamic. It should actually be there. Any molecule can move in anywhere. Okay, subject to the required given conditions of the Brownian motions, which should have be happening, actually happening in the cell. Okay, robustness and sensitivity analysis, numerical simulations, and assessing the results and changing the parameters. All these parts, you need to put it all together in the mathematical model through a computational system. You need to program it. Okay. So data given by is gene ontology, KEG, PDB, Preactome, and all these parts are put together. Here the XML-like annotation languages becomes very important because we need to just download the data in XML and then link it all together. Okay. So the SBML Systems Biology Markup Language is also very important because there also XML is only used. Okay. So information retrieval, we need to develop the tools which should constantly update itself with newer and newer data. With newer and newer information, the tools should be itself able to like keep on updating for yielding the required best results. Okay. And from multiple like remote databases, they should be able to constantly take in the information. We need the system should be able to correlate the data coming from various sources. You need to be able to prioritize which one is more important, which one is not so important. Okay, what is duplicate here, which one is the good data, which one is the bad data. So the program should be really like robust enough. Okay, statistics, data mining, like pattern analysis, all these parts are really complicated. The mathematical scores are worked out at every single step. Okay, and then they are keep on the system keep on integrating them. But this complete analysis or the complete research is very interesting. All those audience who are interested in this part can write to me. We can discuss it, and then you can build up your research profile in this part. It's really interesting. Interdisciplinary information exchange. As I have been saying, the complete part of this systems biology requires interdisciplinary information exchange. So all the parts, the different things are worked out by the scientists, so that the information is always shared. Okay, mathematical formula have to be made understandable for biologists. Okay, the people acquainted with rigid rules of mathematics and computers have to be understand the diversity of biological objects and uncertainty in the outcome of experiments. So you need to understand everything properly to give the required mathematical model. But what are the challenges coming up here? Now see the challenges. I hope you might have already understood. The threshold states, the percolation, like the variables are too different. The variable values, the different gene values, the expression rates constantly keep on changing. There are multiple steady states. It's not binary, 0 or 1. But there are multiple steady states. Even if some protein is getting expressed at a very basal level, very less level, then also it's like non-functional. But still it is there. However, for some proteins, it may suddenly switch to a different one, the functional state, at which it is actually needed. Okay. The activation, the response returns to baseline. Okay. The coating pulses over and undershoot in response. Sometimes the expression values drastically shoot. Okay. Sometimes the expression values remains very low for the reactants. Okay. For the inducers. Okay. And the things keep on changing. Learned responses. See? Long-term facilitation or depression. See? The system is able to learn itself. Next time when the thing come up, it may respond better. Swarm or collective behavior. See? which normally happens in the computational model. The system is able to work it out. So we need to understand that all these parts should be able to properly work out. Okay, the computational model should be able to better represent the biological part. See, this is the next challenge. See, even if the smart therapeutics against drugs, see, if the drugs aren't failing for some diseases, which the system should be able to give us better drug molecules by using all the parts, all the available information. This have shown electronic switchboards to represent different genes coming up. How they are connected among each other, I can show it as like electronic gates. Although these electronic gates and or not, nor and all of them are actually not in mathematical aspect. The meaning intention is for the biological genes. Sometimes some genes are working only when they are present in combination or present either one, either of the two. Okay, or not this or something else. Okay, with that aspect I'm calling it as. Through all these parts, I should be able to integrate all the genes together in a complete pathway to give you the required results, to give the required Boolean table here, 0 or 1, working for me properly, okay, under some required conditions. But it may not be seeming so simple for every case. But yes, people have worked out. Okay, the things quote of like the systems biology, like markup language or systems biology workbench helps us to devise the mathematical model, probably the best model which is feasible for the given conditions, given constraints, and then we are able to devise the drugs, better molecules which is able to interact with several different required targets the best possible way to get us the required results. It should be able to block the required complete phenotype which is working on in the bacteria or virus. Okay, so the drug should be probably the best therapeutic molecule, potential molecule, like better than the ones existing in the market. So it's very useful to design drugs, but it's very challenging. The complete picture looks so simple, but it's very challenging all throughout. Okay, systems biology is an integration of data and approaches, as we have been discussing here. See, it's so much complicated. So, like, what are the major aims of systems biology? See, like the find out the like which molecules try to interact talk to each other in network. Okay, the basic metabolic network, which molecules are actually interacting. So if I'm given the set of interacting molecules, okay, the protein under cell signal transduction or the cell cycle, which molecules are actually interacting, or protein DNA, see the gene regulation among all these parts. If this diagram shows different molecules interacting here, which molecules actually interact together? Okay, how to find out? So the systems biology helps me to better understand the complete part. Okay, the second part, see, determine network topology and the interacting proteins. See, which protein among them? If I say some protein are the most connected to each other, so some protein which is highly connected to each other. See, something which is shown here. See, something which is connected to several different enzymes are the ones which are termed as hub proteins because they are governing several different parts. The term is known as centrality also. Okay, and remember the centrality also. How many times the protein was coming up in the center across multiple different enzymes. Okay, so this shows that that enzyme is really crucial for the complete biological network. Now see the next part. Here, then the tier 2 and tier 3 part is responsible for deterministic and stochastic part. Deterministic, as we have been discussing, if we are able to build the equations properly, we should be able to find out exactly, okay, that if you are changing some sort of reactants, how the product will be changed, and at one point it will change. What will be the rate? But stochastic just means the dynamical aspect, that it may be changing like this. Okay, dynamics is predicted with given initial conditions, although it's not guaranteed to be the same way. Okay, that is termed as stochastic. See, if I'm able to get something, it will be like almost similar. But if the see complete dwindling nature, see the undulations are too much. So now answer these questions if you understood so much. Which of the following best describes the primary purpose of systems biology in molecular biology? To study individual molecular components in isolation. Isolation we are not at all discussing. We are considering all of the molecules together and how their complete network or the complete system will be working out. So the first answer is wrong. To analyze the structure and function of DNA, this is itself wrong. To understand the interactions and behavior of biological systems at the molecular level, this seems to be the right answer. To investigate the effects of factors on gene expression, this is also the wrong part. Now answer the second question. Which of the following is not a characteristic of systems biology in molecular biology? Right? Is not a characteristic of. It is an interdisciplinary approach. It is indeed a characteristic. Focuses on individual molecular components. Is it focusing on like individual molecular component? It is not at all focusing. It is focusing on the complete group of elements together. Okay. Integration of experimental data and computational modeling. This is also correct. Study of complex biological phenomenon. This last answer is also correct. So in this part, the first one, third one, and fourth answers are correct. Only the second answer is wrong. So this is the right part. So as we have seen so far, okay, that systems biology complete part is very complicated, and it always needs the exchange of information among various scientists and be it the researchers working across various fields, wherein there's a big problem of understanding the nomenclature, understanding the variables represented or defined by other researchers. Okay, so only people have been thinking of building a common platform through which they can integrate all their available research the best possible way in getting the required information properly. So, for which Systems Biology Workbench or Systems Biology Markup Language or this programming language like HTML, Systems Biology Markup Language (SBML) have been developed. Okay. So Hamid Buluri and his team have developed this SBML part, wherein you can always computer readable format of various models of biochemical reaction networks. You can always share them, and these are like completely the system is able to recover or read the metabolic networks, cell signaling pathways, the regulatory networks, different enzymes, metabolites, reaction rates, and whatnot. Okay, all the complete information can be easily extracted from these files and can be easily shared as well. Okay, the key features of SBML include modularity, like different modules are there, just like the big programs in C or Python or any big languages. Okay, they are modules there for defining equations, calling some functions, and like that. Reusability. Okay, the SBML part format can be always used and like easily like analyzed through various different tools and platforms. Okay, without much tool like specific guidelines for each and every version. No, it can be properly analyzed by each and every part. All these models can be easily analyzed and modified through various tools, which allow it to increases its reusability. Okay, normally it does not happen that way for other teams. Okay, the researchers, they just make up some image and put it in a research paper. I cannot use that raw data until they give me some raw table file, raw data file like that. But here it is not like this. The math representation. Normally, when we are building up the mathematical formula, mathematical for tuning mathematical models for all the parameters, all the like all the reactants, all the metabolites, all the enzyme parameters. Here, as you can just make up some equation, and that equation can also be very well shared. Okay, compatibility. It is applicable, easily compatible with various tools and libraries. So you can always extend your knowledge among various resources, various software library packages. Okay, see, like this. SBML goes like this. So list of species slash list of species here. Okay, just like the XML was there, body slash body down. And then in between the source code was there for the body. Okay, exactly the same thing works out here. For each starting function, you need to put a down function with slash. Okay, see, the complete part can be worked out. And this systems biology markup language. And here all the tools can be mutually integrated. You can just call some various functions that the libraries can be calling SBML, or SBML can be called by various other libraries. So systems biology complete part includes many components. Okay, cells, tissues, organs, ecological webs of the biological systems being studied. It completely includes all of them. See, to understand the complexity of a problem, a better way it requires a combination of experimental and computational approaches. Okay, all of them should be analyzed together. See, linkages are established between various biological features and systems biology results so that we can get it properly. Computational models of cells are used to like you are built to understand the cellular systems better way. Okay, it is properly needed. Predicting how the characteristics will change in response to the alterations in the environment or system components. Okay, what are the systems biology is able to do? It should be able to predict how the system behavior will change the moment some component constraints or some environment changes. It should be able to understand or predict the required parameters the best possible way so that we can understand the cellular system a better way. See, this part I am taking it from Smith as the like March 2004 paper. See, they have shown this complete diagram. See, so much complicated diagram of metabolic network. Wherein this part is showing the figure 2 of cell division cycle chain CDK, see, along with P21C, different enzymes and how they are mostly connected. And all these are governing the cell cycle under different conditions. See, so precisely it is controlled, and they have done all this. See, so beautiful it is. Putting up so much information together in a single diagram. It looks simple, but it's very complicated. So see the level of complexity existing here. See 2.36 million proteins, the RNA see 270,000. See small molecules, millions, ions, millions, and types like thousands of proteins, like 5 RNA types, 500 small molecule types, 20 to 30 ions, and millions of these molecules are kept always in concentration. Okay, are always kept in concentration. So, such is the big complexity present in a small E. coli, and the system is able to should be able to simulate all of these to get the required part. To understand that the complex biochemical networks always exist in a cell. The behavior of cell is governed and coordinated. See, coordinated by biochemical signaling networks. They translate external responses, all the responses coming up. Okay, hormones, growth factors, the stress, etc., all of them, into adequate biological responses. Okay, such as cell proliferation, specialization, metabolic control or death, or the expression control of several different genes or production of some other key molecules. All of them are governed properly. Okay, but what is the problem coming up? To identify the signaling responses and understand the biological responses for an inducing molecule a better way, so that my system is able to get the required results, okay, and with very high accuracy. That's the entire problem here. The motivation for all this is be like a failure of stimulating responses to functional proteins can be studied better way because it normally causes some diseases, and that's the key motivation which allows us to develop the like better tools. Like it is vital information resource to develop improved drug molecules. Okay, it is highly relevant in discovering more complex relationships involving multiple genes. Okay, for epigenetic effects. Okay, this this may create new opportunities for drug discovery and better medical therapies for the individual treatments as well. Okay, so all these are very crucial, and we should effectively know the complex biochemical networks. So whenever we are saying the biochemical network, it completely like the signal transduction, like it also involves the signal transduction. Protein-protein interaction is one, but signal transduction is also the key part of biological networks, biochemical networks. Here the outside is one primary messenger. Once it passes on like completely part, it initiates a messenger part. Okay, then it goes and initiates some sort of other changes within the like complete nucleic part, nucleic acids. Okay, so because of which the expression of some key molecules takes place in the cell. So see outside if it is insulin, it binds to insulin receptor. So inside the cytosol, what happens? Intracellular signaling works out. Pathways of G proteins, adapters, and kinases work in. Okay, they act as primary messengers. Then after that, it goes and binds within the chromatin, the DNA required slots. Okay, then what happens? And these parts then work out transcription factors and gene expression. The secondary messengers then work out the required part. So small signal insulin leads to many different things. Okay, so but interpreting biochemical networks as concurrent communication systems. Okay, it's not just one molecule. It is leading to the cascade of several other reactions. So the moment in my the computational system, if I'm getting the molecule like this, my computational model should be able to effectively work out the desired set of changes in the computational system as the same such, so that I can get the required results, which should be applicable for the cell, or which should be like there for the cell. I should be getting that. Okay, so biological networks usually consist of concurrent chemical reactions and different molecular interactions. Like RAS-RAF pathway is another thing, which triggers the cell division, and PI3K AKT pathway, see, which keeps both the cell alive and are both triggered by the same growth factor. The growth factor includes RAS-RAF part. It initiates PI3K also, the AKT signaling. See, this is important for survival, AKT signaling. But RAS-RAF pathway is important for cell cycle progression. Okay, so proliferation successful, cell divides. So, so properly like the complete biochemical networks are intertwined. Okay, the interlinking is there, and the complete computational model should be able to interlink all the given models properly. Okay, remember the modularity within the SBML part, modularity of various interconnected enzymes. We should be able to it should be able to completely link all these modules to give me the required results. Okay, see the cancer cell network is much more complex than this, but I'm just showing you a small diagram from the this Hanneberg and Weisenberg 2000 cell paper. For the P53, it's termed as the guardian of the genome. It detects the DNA damage and halts the cell cycle if the damage is detected to give DNA time to repair itself. So the damage equals true and the desired repairable. If the DNA is repairable, it halts cell cycle. P53 halts cell cycle. But if damage is true, damage is there, and it's not repairable. Repairable is false. See, it induces apoptosis, the complete cellular suicide. See, so the same part which actually goes on here can be represented as a program, like putting the required characteristics. Exactly the same thing works out in different conditions. I'm just showing a small diagram or small part here so that you can understand that it can be worked out. Now see the part and answer this question. See, X0 is the initial substrate. It forms six different substrates, S1 to X6, and then finally producing the X1. See, now understand if the V here S3 to S4 decreases to 50%. Its productivity decreases, the velocity decreases 50%. Okay, so if the dV by dt is 50%, the S3 to S4 production rate will go down to 50%. What will happen? Tell me the required answer. X0 and X1 are the fixed states. All reactions are reversible. See, many different answers are there. See, something will be the correct. But the concern is, if I am putting up all this and I ask you that tell me the required answer, what will happen? Based on all these conditions, it could be there from this complete network, which actually there is a cell. It becomes really troublesome. For this complete part, you will certainly give me the required answer. Within some time, you will certainly give me. But like if I'm asking you the answer from this, like anybody can may scratch their head how to work it out. But this is the real complicating situation which the system works out. So once you're able to parameterize the system, the computer should be able to give the answer, even if it is much more complex than this. Okay. Now let us answer these MCQ questions. Okay, if you have properly understood the complete session so far, you should be able to answer these small questions. Which of the following best describes the purpose of Systems Biology Markup Language? Okay, is it a programming language for creating video games? Okay, of course not. Okay, so the first answer is wrong. It is a standard language for modeling and simulating biological systems. Okay, this is a correct answer. It is a markup language for creating web pages. That's an HTML. Okay, and it's a language for designing electrical circuits. So these two are completely wrong. Okay, what is the primary benefit of using systems biology markup language SBML in computational modeling? Is it enhanced visualization of biological systems? No. Okay, because for that all the network analysis tools are there. Okay, simpler data storage and retrieval. This of course not. Facilitated exchange and integration of biological models. Okay, the option C is the right answer. Accelerated computation speed. That is also not the primary goal. Okay, primary benefit of it. Okay, so the option B here and the option C here are the right answers. So now see understand that how the SBML is developed as such as how the SBML programs are encoded. Understand that there are three basic levels of this programming. First one is the most basic compartmental modeling, wherein what happens each cell is considered as a subset of several different compartments, like defined in terms of organelles, defined in terms of biophysical classes, okay, and all of it. The second part includes the mathematical functions and the user-defined functions, conditional events. This defines basically the reaction conditions. So, okay, through which different equations, different parts can be solved better way. Okay, the third one, the level 3 is under development, but this allows us to change the multi-state species, which is normally the case of ions. Okay, the model composition, the submodels, different diagrams, spatial features, and many more. Okay, so this is how the complete SBML levels are there. This how the things are programmed. All right. See the understand that SBML uses a software libraries. Okay, just like the library packages. Okay, so for this, the libSBML is there. This is a specific library only for the SBML. Besides this, for all the analyzing the equations, writing the better like much more sophisticated equations, it also allows the integration of various tools like Mathematica, MATLAB within SBML. For this, the Math SBML is the library used from Mathematica. Likewise, SBML toolbox is the like part modules written only to integrate MATLAB. But these two very sophisticated mathematics tools can be integrated in SBML part. Moreover, there are conversion tools also, the KEGG to SBML, through which any kind of KEGG data can be transported into SBML. Okay, so that SBML analysis can start from the results of KEGG. Okay, so the KEGG results can now be fed into SBML for further analysis. Likewise, the CellML, another tool is there like KEGG, that also can be transported to SBML part. Okay, besides this, there are numerous validation models, visualization like conditions, all the examples of models to analyze various different sources, various different results. All of them can be analyzed properly by SBML. Okay, and they can be all integrated well together. See, to analyze all of this, the the quantitative modeling is the key aspect. For this quantitative modeling, the EBI portal has developed up a server. Okay, the biomodules, the complete functional modules, all these parts allows you to do like complete quantitative modeling of different results coming up, like from SBML and different parts. This is these results are also taken up from different resources like published papers, different research papers, and moreover, the humans properly curate all the data. Means there is a highly accurate data coming up for all the quantitative modeling. And for this, the biomodels attempts to be the sysplot of all these quantitative modeling aspects. Like sysplot data sequences are manually annotated sequence. No, nothing is wrong there. Likewise, the biomodels data are highly accurate. Okay, and lastly, the formats of biomodels are well accepted in SBML and CellML. Okay, so it's a very well integrated tool. And now understand these annotation sources. I'm like enlisting here various servers for doing different kind of analysis. All of you can see this in more details. Okay, see the model part, submitter, creator, publication, gene ontology, ICD-10, pathway, okay, OMIM, the ectome, taxonomy, the compartment part, this gene ontology. Only the gene ontology allows you to like analyze different compartments in which the protein or gene is functioning. Okay, the species part, bind complex, okay, CHEBI, ensemble data, see gene ontology part, and like this. Okay, all of them allows you to do many different things. Okay, the rule, reaction, event. Okay, see now the pathway generation tool. Okay. Once if you have the data, how do you generate the pathway? How do you build up a modeling tool for the biochemical network? Okay, so that you can fetch the data from the required sources, then build the model. Okay, so that you can build a complete biochemical network yourself. Okay, so this CellDesigner is the tool. See? And SBML goes like this, as I've been saying earlier. SBML slash SBML down. See, like this model slash model here. See, annotation. See, the slash annotation comes up here. In between the layout information. See, the complete part you put it as such. So just like HTML, the things are worked out here. See, and here the modelers, all these modeling tools, modelers part, they are not intended to write their models in HTML by hand. They can always get the data from different libraries to work it out. Okay, software tools we read and write the data, and all this is worked out in levels, as I have just explained to you. Okay, and it's all worked out through SBML, it's XML-based language. Systems Biology Workbench is a tool which allows you to integrate the SBML with various other models. Okay, and it supports the resource sharing as well. Whatever the data coming up can now be fed into different other tools. See, this is the SBW part. It provides the C++, Java, Python libraries for Windows and Linux. And it's an application programming interface. It's a highly robust tool to allow you allowing you to do several different analyses together. And many of these tools have been worked out. See, CellNet Optimizer, Boolean Net, Gene Sim, Squared, like that. And there are many more. See, now see the advantage. What is the advantage of doing all this systems biology part? See, understand its part, like how the signaling of all the molecules is worked out in the primary and the transformed hepatocyte cell. That is only the question. See, for the primary part, you see this kind of proteins coming up here. For HCG2, and see the complete part expression pattern is completely changed. See, and see how the difference is there. See, the primary and HCG2. See, these models are substantially different. See, the different connections coming up here. All the connections, okay. All these connections are substantially different. Okay. So when you put up the model together, when you put up the data together, it allows you to build up a complete biochemical network. That this is how the things are working out, through which you can analyze it further. Okay. Integrating the data through various sources together. So, see. So the quality assessment and quality control of PPA data, protein-protein interactions. See, the key problems of the PPA network. See, they're incomplete, they're noisy, they're inaccurate and mixed. See, all these protein-protein interaction networks are always incomplete. Or some of the other protein will always be connected, which we are not able to easily find out. Okay, some unnecessary proteins, trivial molecules will always be coming up, through which some inaccuracy turns up. Okay, some mixed data will be coming up, which I don't want, but still it will be there, which is so difficult to be caught by just like that the different experiments. See, the reason is if I'm taking 10 proteins, understand, If I am saying every protein is interacting with five other proteins or two other proteins, or they can have 2 to 5 interactions, means there can be many interactions, like 20 to 50 interactions possible. Okay, so to find out the 20 to 50 interactions, you need to do the mutagenesis experiments, costing you so much money. Okay, or the SPRS experiments, surface plasma resonance, or the basic chromatography experiments. Okay, whether these proteins colorimetric experiments, whether these proteins are interacting or not. So there are actually so much part coming up. Okay, so many things coming up. And for analyzing all this PPA data, see, different servers are there. See, bind data, biomedical interaction network database, biogrid. See, the DIP database of interaction proteins, the HPRD, the human protein reference database, in Interact, see, the database system analysis for PI data, and the multi interaction database, the Mint. See, all these parts are connected together. So, and for systems biology, we need to analyze all of them together. Then only the things can be properly worked out. Okay, and it has got several challenges. Okay, we no idea how to integrate discrete models. Every protein has got its own model. The moment I take some database, it has got its own result. And the moment I integrate them together, some new result turns up. So I am facing difficulty how to integrate that together, or which part is much more crucial for me. Okay, so no optimal solutions works out. How to fit the data to the model for discrete modeling. Okay, you do not have the correct algorithms for every step in systems biology. So people do numerous steps. Okay, at every step, for they do numerous analysis for every step, then finding out the best feasible part. Okay, new modeling, data analysis, integration approaches, and tools are always needed in the systems biology part. Okay, and thank you very much. If you have got any tools, you can always contact me. Goodbye. Okay.